So I'm really happy to see so many of you, and I hope my talk is interesting to you. So indeed, my field is epigenetics, and sort of the first half of my talk is about epigenetics, and then a bit about the power of scientific research, why it's such a great career, and then about the Institute of Agora, where we merge science and medicine the best we can. Okay, so I'm not really sure how much all of you know about this field, so I'm going to be a little bit didactic. Whoops, sorry, back. These three images are yeast cells, fly eyes, and a calico cat, and they all have one thing in common. I don't know if you know what that is. They are an example of variegation, of epigenetic variegation, because all these yeast cells in each of these colonies, and all the cells in the fly eye, and the skin, or the hair cells of this cat, they're all genetically identical, but because of a random inactivation of genes, they show a variegated phenotype, genes being on and off, even though in a random manner. Now, this happens to all of us. It happens to your patients, especially female patients, because they have two X chromosomes, and one of them is going to be randomly inactivated in the different cells of her body. So epigenetics has an impact on how our bodies respond to the world. It has an impact on everything, basically. Now, variegation is only one part of epigenetic control. So I'm going to tell you, this is the menu for today. Since it's almost supper time, I give you a menu. What is epigenetics and why you should care about it? What defines an organism, nature or nurture? And that phrase, nature or nurture, is age-old, but the question is it programmed? Is it hardwired? Or does environmental impact change the way cells and bodies and organs behave? Why should you be a scientist? And then why biomedical research? So that's the menu. Okay, so to be very simple, genetics, we all know. Genetics is the blueprint of every organism. Every organism, all humans, all cats, all dogs, they have a blueprint to be a man, a cat, a woman, a cat, or a dog. And the replication of that goes on in every cell, and every cell of your body has more or less the same blueprint, some errors, but mostly the same blueprint. And then it's passed on to the next generation. Now, epigenetics is something else. Epigenetic describes how, with the same genome, in every cell of your body, you get tissues of completely different structure and different function. That's because even though genes define all of our organs, all of our program to be a complex multicellular organism, it's the epigenetic control of the genes that allow muscle, connective tissue, nervous tissue, endothelial tissues to be determined and maintained. So epigenetics determines cell type, and it is partly programmed and partly stochastic or influenced by environment. Okay, so this is the subject of the talk. Now, it gets very quickly into the question of memory, because if you have a liver cell and you have an injury, or a skin cell and you have a wound, the fibroblasts reform the skin. They don't form a bone or anything else. Or the liver cells, if they have to regenerate, they regenerate a liver. The stem cells in your bone marrow, they regenerate blood cells. They don't form any other type of cell. So in fact, from a stem cell, you get a progenitor. From a progenitor, you get differentiated cell types. But in fact, at each stage, there's a memory function. And the cell knows that once once a hormone, a growth hormone or some trigger comes, it knows that it will make the cell will make skin or the cell will make muscle, but it won't make anything else. So this cellular memory is, in fact, essential both to growth, it's essential to self-renewal, to differentiation, and even de-differentiation, now that we know that, in fact, you can take differentiated cells, reprogram them back into stem cells, and use them to regenerate tissues. None of this entails any change in the genetic material. It's all epigenetic, as is the degeneration of that program to provoke aging or cancer. So, in fact, epigenetic marks are regulating the maintenance of tissue, the differentiation of tissue, and, as I'll show you, the degeneration of tissue. So how does it work? What is an epigenetic mark? How does epigenetics influence this blueprint which is embedded in the DNA? Well, our DNA, naked DNA, is never seen except under the microscope like this. This is an unfolded metaphase chromosome imaged by EM microscopy almost 50 years ago. But actually, it's never like this. It's always complexed with nucleosomes into beads on a string or folded up even more. And the basic unit, actually, of chromatin and of epigenetics is the nucleosome. The nucleosome is formed of 8 histones. The DNA is wrapped around. But the real information that's contained in the nucleosome is on the N-terminal and C-terminal tails of the histones. I'm sure you know this, but I'll cover it anyway. Their lysine residues are the usual target, and they can either be acetylated or methylated or ubiquitinated, and these modifications become a secondary code on top of the genetic code, determining whether the DNA underneath that's wrapped around this nucleosome will either be accessible or not. So how does that work? Well, a lot of it is electrostatic. In fact, if you have acetylation marks on these tails, acetylation marks are charged, and so they actually open up. They open the chromatin because the acetylation charges don't want to interact with each other, the negative charges. And the methylation charges, they do not change, the methylation groups, they do not change the charge, but they provide a binding site for ligands. So covalent histone tail modifications are at the heart of epigenetic memory, at the heart of cell type memory and differentiation. So we know that nucleosomes in general, because they coat the DNA, if you have a gene that has regular nucleosomes on it, it can be transcribed, but at a much lower rate. And that, in fact, the marks on the histone tails signal either openness for Pol II transcription or a closed structure for repression. So this is generally acetylation that opens nucleosomes, and it's generally methylation that closes it. And in fact, there are typical marks for euchromatin, the open chromatin, and heterochromatin, the closed chromatin. So these are the language, this is the tool of epigenetic control over cell fate. Now, how does it work? I mean, this is in very simple form. In bacteria, you probably learned that, gene regulation, it was the system to learn about gene regulation. In fact, a transcription factor can be induced, let's say on galactose or on a substrate, and it binds the promoter and turns a gene on. And that gene makes RNA, and then the RNA makes protein. And this is called the central dogma of molecular biology. And this is how everyone thought gene expression worked. But in eukaryotes, and that includes humans and everything multicellular, in fact, our genome, as I showed you, is organized into chromatin. And it's not a simple, it's not taken for given that a transcription factor will actually bind its site. In fact, the chromatin can be coated with marks, the nucleosomes coated with marks that repel or prevent a transcription factor from binding, and yet others will be recognized by transcription factors and will activate a gene. In addition, you have RNA coding the DNA. You have RNA also regulating protein expression in the case of microRNAs, and proteins, of course, both as ligands of the nucleosomes and as nucleosomes and the enzymes that modify them. So in fact, things have feedback loops and complexity, and so eukaryotic gene expression is a much more complex question than transcription factor binding. This flexible, conditional expression of genes allows us to make a complex human body and the thousands of different cell types from a single genome that doesn't have that many more genes than a bacterium. A simple yeast cell has already 6,000 genes. A human only has 25,000. So it's barely 4 times more. And yet the programming is extremely complex. Now, this also, the fact that these marks give another layer of control, this is actually the control on which environment, stress, nutrients, and what I mentioned before, stochasticity, can affect gene expression and affect the outcome of an organism. So whereas things are pretty hardwired in a bacterium, of course you can have environmental induction, but then once induced, a transcription factor will bind, it will turn on the gene until something kicks that transcription factor off. In the case of a eukaryotic gene, there are many factors that are going to play into gene expression. So transcription factor binding is controlled. Now, once again, we ask the question, in the development of a complex organism, the formation of all these tissues in our body from a single genome, is this nature? Is it hardwired? Or is it subject to environment and nutrient and stochasticity as we develop? So we wanted to ask this question. This is the major question that my lab addressed over many years. We wanted to know, is epigenetic repression or epigenetic marks, are they required to make a complex organism? Now, to ask that, you have to use genetics. You can't just make correlations. You can say, oh, well, in human cells, there are lots of methylated histones, there are lots of acetylated histones. There's a correlation. But that doesn't tell you it's necessary. It only tells you there's a correlation. So to test whether there really is, we used a simpler organism, not humans. Obviously, it's hard to do experiments with humans. We used the C. elegans, a worm. C. elegans is found, it's a very abundant small organism, multicellular, has only less than 1,000 differentiated cells, and it lays 200 fertilized eggs in its lifetime, which is about 14 days. And it develops from a single fertilized zygote into a larvae who has all of its differentiated tissues in just 14 hours. Now, the worm has basically everything we have. It has neurons, it has a gut, it has skin, it has muscle, it has fat bodies, it has a gonad, and it reproduces. So it basically has the differentiated tissues that we have. So we could use this organism to study whether epigenetic marks are necessary for the differentiation of tissues. In other words, do we need all these epigenetic marks to make an organism. Now, the mark that we studied most intensely is a repressive mark. It's called H3 K9 methylation, histone H3. It's a lysine 9 that's methylated, and it's very abundant on repetitive elements, but also on tissue-specific genes when they're turned off. So this is the classic mark of a repressed gene. And if you think about it, if you start from a pluripotent stem cell or a pluripotent embryo, as you make a tissue, one of the most important things to do is turn off the genes that are not supposed to be on. So we decided to study this conserved epigenetic mark. It's found in everything from yeast to man. Every lysine can have 1, 2, or 3 methylation groups on it, and so we're looking at, a combinatorial complexity, but mainly methyl 2 and methyl 3 on this tail of the histone H3. It's a binding site for proteins that repress, and that's probably why it forms this compact heterochromatin. So we looked in worms, where was this heterochromatic mark, or where was this repressive mark? Now the worm chromosome, there are 6 chromosomes. They're of moderate size, smaller than human chromosomes. The genes are found, this is a distribution of genes along the chromosome. This is chromosome 1. They're scattered all along the chromosome arms. There's no centromere because the chromosome is holocentric. But the repetitive elements are not random. The repetitive elements are extremely enriched on the ends of the chromosome, and so is, in fact, the H3K9 mark. So this was somewhat to be expected. Repetitive elements are non-transcribed, and the mark was on the repetitive elements at the ends of the chromosomes. And this is how you figure that out. You use antibodies that recognize the dimethyl or trimethyl. You pull down the chromatin and the modified histone, and then sequence the DNA that's associated with it. And here we see simple repeats are coded by methyl 2, H3K9 methyl 2, and a transposon cluster here, this is a repetitive element, is coded by H3K9 methyl 3. And genes that are inactivated are much like this. They have methyl 3. So using genetics, we could eliminate these marks and ask what happens. So there are 2 histone methyltransferases. We could eliminate both of them by genetics and then do mass spec and make sure that we were actually eliminating the H3K9 methyl mark. So by doing mass spec on the histone tails, we could show that if you do a double mutant here in the red arrows, you eliminate all the K9 methyl 1, 2, and 3. In other words, we knocked out both of the methyltransferases, and those were the only 2. If we looked by immunofluorescence, so then came the big question, would the worm develop? If we knock these out, that means it has none of this repressive mark that's conserved across all species. And indeed, what we found was that they formed worms. They formed gonads, they formed embryos, they formed larvae. In fact, all the tissues were differentiated and created in the absence of this methylation mark. Now, that's a major discovery because, in fact, we all thought, well, this repressive mark would be essential. So we decided to look more closely because, we did expect some phenotype. And in fact, what we found was that developmental timing was delayed. But the most impressive defect was that the worms became sterile. If you count the number of eggs laid, normally they lay about 200. In the yellow here is the double knockout at 25 degrees, which is, of course, a normal temperature for worms to live outside. There were no offspring. So the worms, we could propagate them at 15 they still laid eggs, but they were temperature-dependent sterile. And when we looked at that more closely, we found that that was because there was massive apoptosis in the germline. So the eggs, as the germline, as the oocytes were developing through mitosis and forming eggs and sperm, they were apoptosing, they were dying. Now, this was provoked by DNA damage, we showed, and the worms without the H3K9 methyltransferase were also extremely sensitive to induced DNA damage. And when we look closely at those worms, for instance, at the muscle, this is the body wall muscle. So these are sarcomeres that make up muscle, just very much like body muscle in humans. We saw that there were signs of degeneration. The sarcomeres were no longer intact. They were degenerated in almost every worm. Now, this kind of tissue degeneration is what happens in aging, progeria diseases and aging of organisms. And so we asked the question, what happens to the lifespan of these worms? And indeed, this is a Kaplan-Meier plot. You've probably encountered it. You start off with a population. You look how quickly they die. Actually, most worms have an average lifespan of about 15 days. That means the 50% are dead at 15 days. And in the methylation deficient cells, they had a shorter lifespan. They died early. Now you can say, well, 2 days, what difference is that? But 2 days out of 14 is more than 10%. So if you lived 10% shorter, you would notice. And in fact, if you got rid of the demethylase that removes the H3K9 mark, they got longer life. They lived longer by about 2 days. So in fact, H3K9 methyl, the conclusion of this study was that the more you have stable heterochromatin, the more the H3K9, the longer you live. Now, again, we wanted to know why. What was going on? They could make all the tissues, and yet they were dying. They became sterile. There was DNA damage, and they were looking like old worms. Well, we looked at what happened to these repeats. So there are many, many repeats in the genome. They are both DNA transposons, RNA transposons. These are foreign elements that get integrated in the genome. And they're simple repeats. That's actually not going to work. I'll have to use this. And there were simple repeats. And this is a heat map. So the blue indicates, sorry, the blue indicates a high-level expression. White is very low, and red is actually repressed compared to wild type. And what we could see is that in the mutant, in fact, repeats that normally carry H3K9 methyl and were repressed in wild type were becoming expressed. Now, this is not only happening in these methylation deficient worms. Sorry, whoops. So I'll just go on. But actually, this is what happens in humans with aging. So some of the markers of aging is actually that you exhaust stem cells, you lose stem cells, that's like the germline. You lose cells senesce. You also, at the genomic level, see increased genomic instability, which we could measure in the worm, and heterochromatin decompaction, and a loss of silencing of repetitive elements. So these have been identified as hallmarks of aging. So upon loss of H3K9 methylation, in fact, what we're seeing is premature aging of the worm and premature aging, paralleling what happens in aging in humans. In fact, this is just to show you, this is a genetic map. You know, our genomes are filled, almost 60% are repetitive elements. either transposons like sign elements or line elements, or simple repeats. And they've mapped the expression level in babies versus the expression levels in octogenarians. And what you see is that, in fact, the level of expression, it's off completely in children and on in aged individuals. So the aberrant expression of repeats, simple repeats, links the loss of this epigenetic mark, this heterochromatic mark, to premature aging and tissue integrity in humans and worms. So remember that if ever, you know they have drugs that inhibit the H3K9 histone methyltransferases, they use them for cancer. But at the same time, you may be actually de-repressing a lot of repetitive elements and wreaking havoc on their genomes. Now, just a quick foray, well, how does this happen? Why does transcription of repeats have this effect? Well, in fact, what we found was that the RNA from the repeats is not exported or processed like normal RNA. It stays in the nucleus and forms what we call an R loop, where the RNA hybridizes to the DNA, and this becomes a roadblock for replication, and it also can create single-strand nicks. So these are called R loops, so you can map them, and you can see in this H3K9 deficient cell, embryo, that there's an abundance of this R loop that we can detect by antibody. And those R loops map to the repetitive domains. So the molecular trigger of aging or of degeneration of cells is actually linked to the creation of DNA damage, damage due to an aberrant transcription of repetitive elements, the binding of that RNA to the DNA, creating a barrier for replication, and increasing DNA damage. So if you ask, what good is the epigenetics? I mean, at least this one mark, the H3K9 methylation, which is the most abundant mark, at least genome-wide. The role of it is actually to maintain tissues in a differentiated state. It doesn't drive differentiation. It isn't even necessary to make the different tissues, but it is necessary to maintain the integrity of the cells once they are formed. But actually, we haven't asked the question, what about gene expression? Is gene expression normal in the absence of H3K9 methylation? Are those worms really expressing the right genes? And so, in fact, it's not just repeats that are de-repressed, but if you look, there are actually about 350 genes that are de-repressed. This is an expression profile. A 0 would be everything compared to wild type would be normal. And this red spot, so each one is a gene that is de-repressed due to the loss of H3K9 methylation. We asked, what are those genes? And I'll skip this. And we looked in the tissue of muscle, particularly because we can isolate muscle from the worms. This shows here in red the body wall muscle of the worm expressing a myosin that's fused to RFP. We can sort those cells after disruption of the worm and then characterize the genes that are expressed, affected either in the presence of, either in wild type, which would have the H3K9 methylation, or in the absence of this H3K9 methylation mark. And again, we see about 300 genes that are de-repressed. Now we asked, what are those genes? You can still make the tissue, so it's obvious that there's something going wrong. And when we mapped those genes, we found about half of them in this differentiated muscle, the genes that got de-repressed, misregulated, were germline genes, totipotency genes. So in fact, to make a muscle, you have to turn off the genes of the germline. And then the other genes that were expressed were tissue-specific genes from a whole array of different tissues. So what was going wrong? You were still turning on the muscle program, but you were not turning off the other programs. And this is actually the job of H3K9 methylation. Now you could say, well, that's a developmental program. Was the H3K9 methylation important during development, or was it important in the differentiated tissue? Now, why is that important? Well, if it's important in the differentiated tissue, then you really should be careful about drugging enzymes that deposit this, because you might lose the mark in an adult. You might cause active damage as you treat the human. So we decided we would allow the worm to differentiate with H3K9 methylation, and then once all the tissues were differentiated, we would turn off the H3K9 histone methyltransferase and ask, do we still see genes being de-repressed in that differentiated muscle? So it's something called a degron. We can attach a degron signal to the histone methyltransferase and trigger its degradation when we want. So we did that after the worms were differentiated, and as expected, the histone methyltransferase was degraded. Now what we found was, once again, about 300 genes were de-repressed, actually a few fewer, maybe about 160. But most of those overlapped with the same genes that were de-repressed when the histone methyltransferase was gone all the way through development. And so what we can say is that, in fact, you need the constant presence of the histone methyltransferase throughout your life. It's not a good idea to treat people with inhibitors of histone methyltransferases, that I can tell you, to maintain the integrity of the tissue. I'm going to skip this one. So the take-homes of about 15 years, science is slow, but they are important take-homes, because we can answer something. We don't know everything, but we can answer something about important and major epigenetic marks. The repressive marks are not necessary, at least in worms, for differentiation of tissues. You can ask, well, how does it work then? It's based on transcription factors, almost like dominoes falling down a path of muscle differentiation. What the histone methyltransferases do is shut off the other programs and maintain the integrity of the differentiated tissue. Without H3K9 methylation, you get random gene expression at a low level and degeneration of tissues. You get an accumulation of damage because you're expressing sequences that should not be expressed. And the other important take-home is that it's important not only during development, but in the differentiated tissue. So your lifespan, probably the integrity of your tissues and of your patients, someday you'll be working with elderly patients. Remember that the integrity of the tissue is determined by epigenetic marks. So, of course, we want to know, is it also true in humans? Now, there's only 2 histone methyltransferases in worms, and there are 6 in humans. So actually, it's very, very difficult to do that experiment. And if you knock out all 6, actually, human embryos or mouse embryos don't grow. It's probably due to the massive expression of transposons and repetitive elements, but actually, we don't know why. But we'll see. I'm going to go on further. Actually, the one thing I will say, if ever any of you get into research, one of the biggest questions, and I regret that I couldn't answer it, one of the biggest questions remaining is what targets, in a tissue-specific manner, what targets the H3K9 mark to the sets of genes that need to be turned off. It's easy to imagine transcription factors turning genes on, but we still don't know how the cell knows which sequences to turn off. Okay, so that was a foray into my science. Now, in the next 15 minutes or so, I'm going to convince you you should be a scientist. Okay, and I'm going to hopefully convince, maybe the science didn't convince you, but you can obviously tell that I love my own science, and that's something that's typical of scientists. First, I want to introduce you why to be a scientist, and then I'm going to say why biomedical science. You're already studying medicine, so you can say, well, I've made my choice. But remember, medicine is dependent and is driven by scientific knowledge. So why pursue a scientific career? And these are all the reasons not to, okay? First of all, scientific research isn't for everybody. I have to admit, you'll be not very well paid. You'll earn more money as a doctor. You will work longer hours than anybody else, and there will be moments of extreme frustration because lots of experiments don't work. We only show the ones that work, but there were, you know, years and years of experiments that didn't work. And in fact, to succeed, you need skill, patience, money, and luck. In German, Geschick, Geduld, Geld, und Glück. And in fact, you can't even control those things, you know? And so maybe the skill and patience, but not the money and luck. So why in the world would you be a scientist, okay? Well, I'll give you 4 reasons, actually 5. First, science allows you to stand on the shoulders of some of the most brilliant people who have ever been on this earth. Science is a continuum of knowledge. As we learned for centuries, we've learned about human systems, about molecular systems. Since the 20th century, we have had an exponential increase in our understanding of biological and living organisms. And you can stand on these shoulders and see things that even these brilliant people couldn't see. And if you don't know who these people are, you should. Second, you'll be able to fulfill the most basic drive of being human, which is curiosity. Now you'll say, oh, medicine, I'll help mankind. But in fact, all of us, all humans, desire to know. Plato said that, but it is actually true. And curiosity and finding out answers is the most rewarding thing, at least that I know of doing. And as said by Peter Medawar, science at all levels of endeavor is a passionate enterprise, and the pursuit of knowledge is a sortie into the unknown. And this is actually exciting. Third, you will inevitably discover something new. You won't just be a mouse running in a wheel. You will discover something that nobody else knew before, and perhaps it will be big. And as a medical researcher, you will probably help a lot of people, a lot more with your knowledge than with your hands. Just by virtue of the fact that you're experimenting and seeking truth in an area that no one has explored before, it would be unthinkable that you would not make a new discovery. And there is plenty to discover. If you're always doing new things, there's very little competition, remember that. You can always discover new things. And as a scientist, you will undoubtedly love your job. Because no one undertakes scientific research unless they love it. And actually, it's a completely rewarding job because it's every day is something new. Every day you are defining your own day. So science can be the most enthralling enterprise possible. It gives us the thirst for knowledge and the means to get there. So what should you do to pursue a successful scientific career? Here are just 5 hints. 1, be audacious. Think big. Have ideas that nobody else had. This is actually the essence of scientific research. Challenge the status quo, but stay modest in face of facts. Anyway, scientists are always being proven wrong, so they're usually pretty modest. Use first principles in your thinking. Ask why relentlessly and let science and logic lead. Never do anything just because people tell you to. Find an inspiring mission for your research and focus on it. Be empirical. Don't just theorize, but let the results lead you. And team up with others as you pursue your research. If you're an MD researcher, team up with engineers or with basic scientists. If you're a theoretician, team up with engineers. Collaborate. These are the best advice I could give to anyone starting a career in science. And some practical hints: you need to be ready to move to another country, because seeing science in another environment is very revealing. You should get your PhD and postdoc, or do an MD PhD. If you've done an MD and then do an MD PhD, you actually have done like an MD PhD postdoc as soon as you can, but enjoy the research. Find an advisor you admire but can argue with. Change fields at least once. Don't ask for and don't expect security, financial security. It comes far too late. And find the right partner, because scientists, people who do lab research, tend to be very poor marriage partners. But on the other hand, we all manage. So that's my plaidoyer, or my heartfelt plea for science. Science is actually one of the best careers you can imagine. But why biomedical science? And this comes back to Agora. So Alicia mentioned that she visited this institute. This is the institute that my foundation built in Lausanne, next to the Alps, next to the Lake of Geneva. And in this institute, we do translational cancer research. And the goal is to bring discoveries, basic discoveries in biology and engineering, to medicine. The idea is, in fact, to unite the insights from geneticists, bioinformaticians, chemists, immunologists, informaticians, along with biologists, and deliver insights to medical personnel. Now, how do we do that? The only way is to actually work together. So in this institute, 1/3 of the group leaders are led by clinicians. So these are MDs that spend half their time in the clinic, half their time in the lab, and most of them have labs of their own. And so this Agora brings together 2 hospitals, 2 university hospitals, and 2 different universities, to allow people to explore by combining their expertise. Nobody can master everything. In fact, the problems generally come from the clinicians, and the tools to explore them from the scientists. So what do I mean when I say translational research? Now, discovery research is like what I was telling you about with the worms. we were trying to find what are the pathways that control growth, control cell determination, control aging, and in fact, even lead into cancer. This is actually what's usually done at universities or basic institutes. And then there's clinical research in the hospitals, where they are taking a medication that's already approved and testing it on different subsets of patients, directly working with patients. They're trying to identify, stratify patients, understand comorbidities, combination therapies. This would be what I would call clinical research. Now, translational research is everything that connects these two worlds. The brilliance, or let's say, technical insights of the scientists need to be able to influence the practice of doctors. Translational research is that interface where, working together, one can explore novel targets on human tissue, not using model organisms, screening for novel inhibitors, optimizing treatments, identifying diagnostics, and then understanding the mechanisms of drugs. This is actually the biggest gap in all my career that I've ever seen, is actually the transmission of knowledge from basic to medical practice. Now, this guy, Sidney Brenner, was a Nobel Prize winner, but he did many brilliant things. And he said, progress in science, but you could even say progress in medicine, depends on new techniques, new discoveries, and new ideas, probably in that order. And I actually agree with him, because I told you our knowledge, the increase in scientific knowledge of relevance to medicine has increased dramatically in the last 15 to 20 years. And it was at every stage enabled by a new technology. Now, I've listed a number of technologies that when I was your age and studying and doing research, I had none of these at our disposition. We knew what DNA was, and we could clone it because we had restriction enzymes. But it was very difficult to sequence, just small bits. And in fact, you know, we could not manipulate the genome. We had to screen for mutants. Now we have genomics, deep sequencing, human genomes from single cells. You can analyze every cell in a tumor. We have CRISPR-Cas9, which designs mutants in mammals. You can do this in human cultivated organoids. We have GFP and live imaging of proteins and molecules within the cell. Proteomics, mass spec has improved sensitivity so we can understand all the proteins of a cell. Induced pluripotency, cellular reprogramming. I mentioned that this has to do with resetting of a cell, differentiated cell type. Organoids and synthetic organisms allow us to study human tissues and even tumors in vitro. Machine learning, of course, applied to complex and diverse that this is, of course, even should underlie almost all the advances, because the data that's produced from all the others will be fed into AI. Cryo-EM is another advance, and even just the use of RNA, being able to deliver RNA to mediate gene therapy, is a tremendous advance. And the medical impacts are enormous. However, we have to bring these scientific insights into the proper realm of use within medicine. And as you go forward, and perhaps, I hope, these techniques will be integrated, you will sequence the tumors. You will analyze and alter patients both with fluorescence, with designer proteins. You'll make synthetic organs. You will perhaps even design therapies based on human tissues. So the technological challenges and priorities in our own institute are, in fact, to advance the single-cell sequencing because it allows us to understand individual tumor cells as a complex system, using tumoroids and organoids in vitro so we can study human tissues in a dish and not as single cells, which actually are not really reflective of a tissue. And then, of course, the integration of AI and computer machine learning into all of these technologies. So no one moves forward anymore alone. We work as teams. And we hope the questions come from and the clinical challenges come from the doctors. That's the hospital there behind. And the solutions and the explanations from research. So if in 30 years you're still a medical doctor, which I hope, and you think about research, how it has influenced your practice, you will want to know how these mechanisms work. Because without that, you cannot appropriately apply them. So my encouragement is actually, like Alicia said, do an MD, PhD. After your MD, go for 2 or 3 years to a research lab. Learn what it means to do true experiments, controlled experiments. Learn what it means to do single-cell sequencing and analyze it by AI, or analyze histological samples, not with eyes, but with a computer. This will change the way you do medicine. So my encouragement is go for it. Go for science. Integrate science into medicine. This is actually, you are on the forefront of the biggest change in medicine that has ever happened, I can guarantee. Okay, so you're supposed to scan this. But I'm first going to thank my lab. This is a reunion of ex-lab members over a lot of time. I've been in almost every university in Switzerland, and I have to thank many people for support. But above all, I'd like to thank all the students and MDs that I ever trained, and I hope I can inspire you to actually embrace science and bring it into medicine as you go forward. Thanks.